All right, so can you all hear me? No, reply. I can hear echo somewhere. Bonjour tout le monde, bienvenue à tous. Welcome everyone. I hope you're all doing fine and healthy. So this seminar will be in English as usual for Montpellier Seminars in Ecology and Evolution, aka the SEM, which is supported by the Labex Cemeb. So this one is the first of a series of online seminars or so-called webinars that we have decided to launch after the lockdown has been announced here some 10 days ago. And for this first one, we have opted for something a bit special since we realize we're all a bit obsessed by the current pandemics and try to understand, make sense of all these graphs and curves we see and this complex information we receive from everywhere. We thought it would be nice if we asked some colleagues from Montpellier here to help us navigate through all this jungle. So we didn't ask them necessarily to present some hot new results from their own research, rather some elements of thoughts, or perhaps a digest of ongoing work worldwide, which they will analyze with their expertise in different subfields of ecology and evolution. So we'll first hear Mircea Sofonea. He's from the ET Nivegex, so he'll present some he'll explain the meanings of different parameters of the epidemics, and he'll present work they've been doing their team to try to estimate them in France. Then Nicolas Bierne from the ISEM will tell us about what is known so far about the genomics, the phylogenomics of the virus. And lastly, we'll hear Sylvain Gandon from the CEF, will present results from a simple model he's been working on lately. He's currently working on actually to try and project the possible outcomes of different control strategies that can be implemented. And now I'm going to leave the microphone to Emmanuel Fraunhofer, who will explain some technical aspects of how the seminar will work.
Yes, hello everyone, and welcome also from my side. So as Luis was saying, it's the first time we do this, so please excuse all the bugs that are going to occur and bear with us. We're trying to do our best to keep this going. So as we said earlier, please don't unmute your mics or mute them if you have done so, and switch off your cameras so that we don't risk to overwork the network. Your questions, you can ask them via the chat. So if you look at the lower toolbar, you can activate chat or converser. You can post your questions continuously and they will be answered then at the end. And so don't forget also to say to whom the question is addressed. If in the end your question is not represented correctly or if there's anything urgent, you can raise your hand in the chat and we will try to turn on your mic depending on how much time we have and how many people raise their hands.
Sorry, it's not in the chat that they can raise their hand, it's in the list of participants.
In the list of participants, there you are.
You click on participants and then you have a list of participants and then you have something which says lever la main or raise your hand.
So once the speaker has shared their screen, as is currently the case, if you want to exit the full screen mode, you can just toggle to the upper toolbar and press on exit full screen mode if you prefer to do that. And if you have any technical questions, just ask them via the chat or send us an email. We'll try to answer all of your questions. And of course, if you have feedback, please send it to us. This is a test. We're trying to do our best, so we're happy for any ideas. And now, Mircea, please.
Hello, everyone. Thanks, Luis. Thanks, Emmanuel. The ETA team from the Nivegex lab is currently working on several modeling approaches of this COVID-19 pandemic. And we were asked a few days ago by the organizers of this webinar we thank to expose the principles of estimating parameters of an emerging epidemic in the early times of an outbreak. So importantly, this is not the work we've been doing so far in the team, but just opening the black box of a modern inference epidemiology applied to this pandemic. This pandemic that started from China, as you all know, in mid-December. The first cases of pneumonia of unknown etiology triggered a surveillance mechanism that was built after the SARS-CoV-1 epidemic in China in 2003. And then that allowed the Chinese CDC to investigate clusters of cases in Wuhan and outside of Wuhan. And by doing contact tracing, they could estimate the key features of this disease and also its epidemiology. And this led to this paper published in the New England Journal of Medicine, one of the first to estimate, especially the R0, the basic reproduction number. You know, this is the most important quantity when investigating an outbreak. The premises are traced back to Fibonacci and Euler, so fundamental problems of demography. The notation comes from the seminal work of Dublin and Lotka in 1925. That was a population dynamics problem, and the modern and broadest definition was given by Dickman et al. In 1990 as the spectral radius of a next generation operator that has to be found for each model. So there is a recipe for that, but there is no general formula for that. And this can be seen as interpreted in the first place as a lifetime reproductive success. Applied to epidemiology, a raw interpretation for interhuman transmitted respiratory diseases would be that the R0 is the transmission rate times some duration of infectious periods on time that has to be defined, and susceptible density when the transmission is density dependent. Well, this can be seen therefore as the initial average number of secondary cases. Importantly, R0 is a dimensionless number. This is not a rate, and as such cannot be used alone to infer a speed of an outbreak. What is important and why it is used is that extinction is avoided or avoidable if stochasticity is allowed, if and only if this quantity is greater than 1. So this is used as an epidemiological threshold, the potential of a disease to spread in a population, but is not a measure of the speed. For further reading and perspectives in the 3 fields of population biology, I recommend you these 3 papers in the footnotes. So the simplest way of deriving the R0 is to use the classical SIR model introduced by Kermack and McKendrick in 1927, where demography is neglected and we investigate the dynamics of a single epidemic wave that vanishes with time. We consider 3 kinds of compartments: the susceptible individuals that are not infected yet, the infectious individuals that are infected and contagious, and the removed ones that can be either dead or recovered and immunized. Beta is the transmission rate and gamma the recovery rate. These 3 densities start as follows, where we consider a negligible fraction of infected individuals in a sea of susceptible individuals and no removed individual at the beginning of the epidemic. Then the densities satisfy this system of 3 ordinary differential equations. And by factorizing the gamma, which is the recovery rate in the second ODE, the R0 appears as following this formula: beta S0 over gamma. And it is straightforward that the density of infected and infectious individuals increases with time if and only if R0 is greater than 1. So this increase is true for the starting of the epidemic. Kermack and McKendrick applied this model to a plague epidemic in India, and the model fitted quite surprisingly well the data. As you see, the beginning of the epidemic, if we follow the density of infected individual, has an exponential trend. This can be seen quite easily by integrating the second ODE, assuming that at the beginning, at the early times of the outbreak, the susceptible density is not much affected by the disease. Therefore, the density of infected individuals, I, is a linear function in time when plotted on the log scale. From this simple linear regression, one can therefore have an estimate of R0 given by the estimated slope times an average infectious period, that is 1 over gamma, plus 1. Here I did the regression for the COVID-19 epidemic in France from late February to the lockdown date. However, the R0 cannot be estimated without having an estimate for the average infectious period. The problem is that in the COVID, so the SARS-CoV-2 disease, this infectious period is difficult to assess. What we see is the clinical status. That is, the illness occurs after an incubation period. But the dynamics of the epidemic are governed by the productivity of the infection, that is viral shedding in this case, which occur after a latency period that is known now to be shorter than the incubation period. Therefore, we have additional stages that appear, the latent period E and the asymptomatic infectious E. So this is not the SIR model anymore. Plus, it appears that viral shedding isn't uniformly, has not a uniform distribution over time. Therefore, the infectious period is difficult to estimate. And we have plenty proxies for that, either the time from onset of the symptom onset to hospitalization or onset to death. Also, the duration of positivity of PCR assays, but also the negativation of some subgenomic viral RNA, and also depending on the publications and the countries, these estimates are quite different. This discrepancy in what should be a good average infectious period is quite large, and therefore leads to a broad interval of estimates for the R0 from 2 to 7, which is not suitable if used to implement public health measures. The way to circumvent this problem of defining and estimating the average infectious period is to consider the ecological point of view of the disease. That is a sequence of loops of the pathogen life cycle. The pathogen life cycle is comes from an infector to infectee and so on. The time between two contamination events is called the generation time G, but this is rarely known as there is no sign of contamination, and because there is this incubation period. Rather, one can see the beginning of the illness, that is the symptom onset, and the between symptom onset interval is called the serial interval S, and this can be assessed if one identifies transmission chains, but this is subject to multiple exposure issue and has to be treated statistically. So S is a good proxy for G because the expectations are the same. but variance of S is greater than variance of G. So using the serial interval as a proxy for generation time is possible. One has to do contract tracing. This has been done in the paper I already mentioned by using some, identifying some clusters of transmission in Wuhan and outside of Wuhan, and estimating the distribution of this serial interval. Better estimates were done later in February and found a median of 4 days for this serial interval distribution. This is, in average, shorter than the incubation period. Therefore, this is the evidence that the disease is transmitted partly within an asymptomatic period. Then how to use this data to infer a proper R0? Let's start from a simple, only for the secondary cases, you have one infected infectious individual. You have one infectious individual that can transmit to number Z of secondary cases the disease. But as this is a count data, one has to assess the noise on this data. Therefore, Z is distributed according to a Poisson distribution of expectation R0. Once Z is drawn from this distribution, the times at which the contamination occurs are distributed according to discretized non-exponential distribution, Weibull or gamma or log normal, fitted on the empirical serial interval data. Now, because you can have tertiary cases and so on, each infected individual contributes to the epidemic in a shifted way depending on its generation time distribution superimposed over the time over time from the beginning of the outbreak. Therefore, you have this stacking process that occurs with the YT leaps are the daily incidence, and the sum of these daily incidents are the cumulative incidence, the data that we can have from the authorities. Using data vector of daily incidence, so for each day you have the number of how many people were infected or confirmed cases, and using seeing those leaps as Poisson distributed variables of intensity lambda t that is parameterized with all the daily incidents of previous dates, weighted by the probability distribution of the generation time. Hence, the R0, the estimation of R0, is the value of the parameter that maximizes the log likelihood of this count data. The estimated R0 is therefore much more precise, with a confidence interval that is quite narrow, and is estimated for this epidemic in France about 2.5. Of course, this assumes that everything is the epidemic spreads into an homogeneous population without any structure of age or contact or space. But interestingly, this has been tested by a theoretician. What we use implicitly by using this inference or the SIR model is that everything is under a mean field assumption. That is, everybody interacts with anybody with the same probability. This is a mean field at the network on the left. A true network would be that the number of links or edges in the graph are distributed. And so you have heterogeneity in terms of contact. You can also have a multi-type network where you have differential interactions between individuals at different rates. This theoretical paper applied several configurations of networks and multi-type and compared the R0 estimated with the true data to the mean field assumed model. And the results are that the R0 in mean field over the true R0, and that this ratio is not very far from 1. So, and is usually greater. That means that the estimated R0 based on mean field assumption are more conservative than suitable for public health measures. To sum up, R0 is a key estimation in emerging epidemics to assess the potential of spreading. But reliable estimates requires the serial interval distribution because there is no way of having a good estimate for an average infectious period. The good news are that simple and structured models lead to quite conservative control effort targets. In the end, you can also see what we are doing so far about we estimated using these techniques the basic and also effective reproduction number in France. We discussed the difference between the final source proportion, the proportion of people that are infected by the end of an epidemic wave compared to the herd immunity threshold that can be targeted by public health measure, and implemented the SEIAIR model suitable for this disease. And we're also going to produce some work on fire dynamics, optimal control, and non-Markovian simulations. Thank you very much.
Okay, thank you, Mircea. So I think we're going to switch directly to Nicolas now, who will give us a clap here. There are some claps on the chat. So, Nico, c'est bon?
Bonjour à tous.
Hello.
Hello, everybody.
So some people have asked whether the speakers could use their mouse to show what they would show on their slides. That would be really great.
Okay.
And for everyone else, thanks to Alex Courtiol, we have a really great site for asking questions. And I've shared that link to everyone and I'm resharing it. So if you want to ask questions in a more fancy way, you can just use that website and vote for which is the most important question.
Yeah, and also perhaps make it easier to navigate through because there are many things on the chat already. So.
Okay, c'est parti.
Okay, can you see the slideshow?
Yeah.
Yes, okay.
Yes.
So hello everybody. I am quite an outlier speaker today because I'm really not a specialist of virus. I agreed to give an informal talk following the pressure of the seminar organizers. And you should take this talk like a coffee break discussion more than a true scientific talk. So I'm going to present what have been done with genome sequences. So I'm used with the study of genome sequence, but not so much with virus. The only thing I do that legitimate that I speak today is that I am studying clonally propagating parasite, transmissible cancer. So for 2 years now, I follow the literature. So I was aware of what people in genomic epidemiology were doing. But I've seen in the list of participants that many other participants could have given the talk rather than me. So I apologize in advance. I use this picture for the first slide. It is drawn by Alex Kagan, and it illustrates people in the genomic epidemiology field fighting against the virus, and I like this picture. So what I am going to talk today is mainly comes from the Nextstrain initiative, which is a website with an amazing tool to follow in real time the phylogeny and the spatial sampling of genome sequence of pathogens. You have all the participants of this Nextstrain initiative, and if you want to follow them, you can, for example, follow on Twitter Trevor Bedford, the leader of Nextstrain, the Nextstrain account as well, and Emma Hartcroft, who was depicted actually in the first slide here. The other source of information comes from this website, virological.org, where people post early discussion papers or preprint, and it's very useful to follow in real time what happens. And you can follow, for example, Andrew Ramboat, Tanya Stadler, and Richard Neher. So what can we do with genome sequences? We can first try to identify the species of origin of the zoonosis. We can try to date the emergence. We can try to track migration and detect community spread. And I put cryptic in parentheses because often we can detect migration and spread that was undetected with standard tracking of people with symptoms. We can infer some useful epidemiological parameters, and we can also study the evolution of the virus. So what is really amazing with this epidemic is that the genome sequence of the new virus was available as soon as 11th of January. It was, everybody became aware of this thanks to social networks. And you can see here that the sequence could have been used already at this time. It was also announced by the World Health Organization. And as soon as a few days after, the 14th of January, we already knew that the closest available genome was a bat genome, and that some other sequence from the pangolin were also close to this new genome, suggesting the virus came from the wildlife, especially bats, because it was known that coronavirus are abundant in bats, and the previous SARS-CoV-1 already was found, a close relative was found in bats. For SARS-CoV-1, they found an intermediate host, which was a civet. And for this one, SARS-CoV-2, it was debated if pangolin was a possible intermediate host or not. You can see in this tree that it's not really a good candidate because the bat sequence is much closer. So the Nature genome papers appear a few days later on the 3rd of February. And you can see here the phylogeny. So it said exactly the same thing, that the bat, the sequence from the coronavirus of a bat is the closest relative of the one found in humans. And so why do we talk about pangolin in the media? The reason is that some pangolin sequence were available, And something interesting has been observed, which is that if we focus on one gene, the S protein, and in particular the receptor binding domain, you can see here that the closest sequence, the sequence with the more similarity, is the pangolin sequence. So one hypothesis was that SARS-CoV-2 could be a recombinant between a bat virus and a pangolin virus, and that the pangolin sequence helps the virus to switch to a human host. However, it's not possible to rule out convergent evolution, and you can see here the synonymous tree, which use only synonymous mutations at the same gene, and you can see that the bat sequence is closer to the SARS-CoV-2 sequence, meaning that if you only look at neutral mutations of the gene, you find this phylogenetic relationship. So we cannot conclude if the similarity with the pangolin sequence is due to recombination or convergent evolution. But overall, the virus is more similar to the bat virus than the pangolin virus. I suggest that you can have a look at this paper published in Nature Medicine, the proximal origin of SARS-CoV-2, by these authors, which explain very well what we can learn from the genome sequence about the origin of SARS-CoV-2. Okay, now we can also date the emergence of the virus and the starting of the epidemics. So to do this, you simply have to look at the viruses that have been sampling in time. So you have here the first samples, which were used to make the first genome sequence, and all the genome sequence that have accumulated through times in January, and February. And you see that the divergence to the root of the tree increase with sampling time. And with a simple regression, you obtain the molecular clock, which is around 2 mutations per month and per genome. And you can date the most recent ancestor. So it's not really the true date of emergence, because you can lose lineage during the initial period of the switch to the human host, but the most recent common ancestor is dated at early December or late November. You have the posterior distribution of this estimation here. And here you have the tree. So you can see that there is not a lot of mutation. The molecular clock is only 2 mutations per month, but it's sufficient to have a good estimation of the date of the most recent common ancestor. And what is really awesome is that you can do this with Nextstrain in direct live. So I really advise you to go in this website, Nextstrain, and play with this tool. It is really beautifully done. You can change a lot of parameters. And as soon as a new sequence is released, all the graphs, all the phylogenies are calculated again. And so here you have the new regression that really shows that you have 2 mutations every month on average between sequences. Okay. So Nextstrain can be used to track migration and community spread. So here you have the tree of yesterday, but you can remove the most recent samples. So here I show a tree where you only have Chinese sequence and all the sequence of people that were tracked to come back from China. So it was in January and early February. So in blues, you have the Asian sequence, and in orange and green, the European and American sequences. French were very fast to release sequence in this data set. So, and here in Nextstrain, you can have the information, all the purple dot are people that were tracked to come back from Asia. So at this stage, all the sequence from outside Asia were really from people that came back from a trip to Asia. But you can see here that with the new sequence, you have some clades that were hidden and unsampled at this time. Okay, so if we look in the more recent past, in February, then you have the first sequence from America here in Washington and from Italy here that suggest community spread, so that you have the epidemic starting in Europe and in America. And so I take the example of Washington that is really useful. You have this sample from this person, Washington 1, who came back from China, sample in January. And here what happened is that people in Washington studying standard flu, they decided to test their samples for the SARS-CoV-2, and they found positive samples and immediately sequenced the genomes and obtained this sequence, Washington S2. And it shows a direct link to Washington 1, suggesting that during 1 month, the virus spread locally in Washington and mutate 2 times. You have 2 mutations between these 2 sequences. Okay, and here it's the explanation by Trevor Beresford. So you have this Washington 1 sequence, and during 1 month, you had 2 and even 3 mutations before the sampling of Washington 2 sequence. So here is the new tree. I take the picture yesterday night. In orange, you have all the sequence from North America. In green, all the sequence from Europe. And you can see now that all these sequences are related in Europe. So it's a big European cluster here with some sub-clusters. And you also have other clusters here. So the diversity of the virus in China was completely imported in Europe. And here you have in orange a cluster from North America, mainly the Washington state. And you can see all the sequence with another color that corresponds to migration between continents. Here I show, I have to go quickly, but I show the tree with the divergence, and here it's the inferred tree with times that make use of the information that we have the sampling times of each sequence together with the genetic relationships that allow to obtain the topology of the tree. Okay. And here I focus on France because maybe you are interested by French sequence. And I change the color to have the location of the sequence, not only the continent or the country. And you can see that the sequence are found in every location, in north of France, east of France, in Rhône-Alpes, and it really means that the virus was moving a lot during January and, well, end of January and all the months of February. Okay, and now we have 69 genome sequences from France. So we have to thank all the lab, especially Institut Pasteur, who sequence all these genomes in real time. Okay, so with genome sequence, we can also infer some useful demographic and epidemiological parameters. So basically, we use coalescence theory. Here you have the standard coalescence where the population size is stable in time. Here you have a decreasing population, like a bottleneck, and here an increasing population. The result is that where you have an increasing population, you have long terminal tips and short internal branches. In a bottleneck, it's the reverse, short tips and long internal branches. And in the standard constant size coalescence, you have exponentially distributed coalescence times. So with the shape of the genealogy, we can infer some information about the demography of the population. And in the case of virus, it's often an exponential growth. And we have all the theory to fit this exponential growth, how the population size increase with time in generations. And this parameter R can be used to deduce useful epidemiological parameters, such as lambda, which is becoming an infectious rate, which is a reverse of the duration of infection, and lambda is the transmission rate. So if you have an idea of the duration of infection, the previous talk explained that it's difficult to have, but in the case of SARS-CoV-2, we can use between 10 to 20 days, and the estimation are really similar. Once you know the duration of infection, you can infer the famous R0 parameter, the basic reproductive numbers. So we have now a very sophisticated method. I do not enter into the details, but just provide the results. So it's from this paper. So it's not an official paper. It's in the website I provided to you. It's written by the Tanya Stadler groups. And they use sequence from China, from the Diamond Princess, the ship, from Washington State, and actually Italy. It's not really Italy, it's Europe. But as I explained, Europe is like a single big population. The boundaries did nothing on the virus movement. And they applied these techniques to infer R0, and you can see that the estimation is basically around 2 and 3, and the best estimate is 2.5, 2.7. So similar to the estimate obtained from standard epidemiological studies. And from this data, you can also have the dynamic of the population that can be translated into a count. It's quite small, I'm sorry, but and what is interesting is that you have here in red the number obtained from through test, and always the estimation is higher. It is about 10 times higher. So for example, in Italy, where the number of positive cases were around 2,500, the estimation from genome sequence was 10 times higher, which meant 25,000 people infected by the virus. What is interesting with the diamond princess is that you have a quarantine in the middle, and you can see the change in the demography before and after the quarantine, and also R0 is different. It was higher before the quarantine and after the quarantine. And quickly to finish, some word about molecular evolution. Maybe you are aware that a paper talk about a mutation, an amino acid mutation that identified two lineages, the L and the S lineage, according to the amino acid. And quickly, it was debunking in the web because it can simply be interpreted as an early mutation at the beginning of the spread in China, and there's no real evidence that one of the two lineage is spreading faster than the other or have more effect on the symptoms of people. So it's another example that Nextstrain is really an amazing tool because you can color. Here I have colored the tree according to the genotype, and I just selected the nucleotide position 20,144. It's the position of the mutation, and you have the tree like this. And here, Trevor Bedford show how the frequency of the two lineage vary with time, and you see that the two lineage have the same frequency with time. Here,
Nicolas, are you soon done? Because I think
the last slide
for the conference.
Okay. So here, well, I can this slide just to remember that to date there's nothing really clear about molecular evolution, and all the virus are similar really, and actually this lineage is more frequent in North America, and this lineage is more frequent in Europe. Okay, and I can stop here, and thank you for your attention.
Okay, so this was thanks Nicolas. Can you hear me? Yeah. Thanks. This was really interesting. So that's why, I mean, we're running a bit late, but I think we're all quite interested in hearing about this. Quite a dense digest. I think you were being a bit modest because I don't think many of us could have extracted all this information so quickly. So thanks again. And okay, now I'm just handing the microphone to Sylvain Gandon, who will tell us about the modeling work he's been doing.
Sorry, Sylvain, before you continue, a quick comment. We have, thanks to Alex Courtiol, a link for questions, and I'm reposting it. So if you want to use that for questions, that would facilitate everyone's lives. You can also vote for which question is the most interesting one. Yes.
And I will also encourage the speakers to look at this link. So I sent it to you by email so that you can also anticipate your answers to the questions or even pick your questions if you want. Okay, Sylvain.
Okay, hello everybody. So I'm going to present some of the work that we've been doing with Sébastien Lyon and Troy Day for the last 2 weeks. On the epidemiology and the evolution of the virus. So I guess the work was initiated from the frustration basically that we don't know much about this disease. So one idea to try to get some intuition about what could come next was to try model the dynamics of the infection with very simple models. So maybe unlike what other groups have been doing and are currently doing, which is trying to build very detailed description of the life cycle to try forecast what's going to happen. We've been focusing on a very, very simple toy model which would basically help to build up intuition about these dynamics. So there's two parts in this presentation. The first one is focusing on the possible consequences of the type of measures that have been put forward, like social distancing, on the epidemiology of the disease. And the second part will be even more speculative, maybe, which is going to address the question of the evolution, the possible evolution of the disease. So I will start recalling what Mircea just described before, which is the SIR model, which is a simple description of the life cycle of the disease, which starts with a population of susceptible hosts that become infected at a certain rate, which depends on the transmission of the disease. And those infected hosts can recover and enter the recovered fraction of the population. So the model is assuming that the total population size is constant. And what we're going to look at is the effect of social distancing. So a first effect of social distancing is to basically decrease the transmission rate, so which is going to be quantified by this parameter C beta. So the higher the C beta is, the lower the transmission is going to occur between individuals. And there is another potential effect of social distancing, which could appear via quarantine. So if you quarantine people, it means that you get some people that are infected and you basically extract those infected individuals from the population. So you basically enforce this rate of recovery, even though they may not be recovered yet. But quarantining is going to affect this rate of recovery, and it will increase the gamma parameter. So the basic reproduction ratio of this simple model is written like that. So you can see directly the effect of social distancing via transmission or via recovery. So having this model, you can play with it, you can do simulation. So here I'm plotting the prevalence, the dynamic of the prevalence in red. So this is the density of infected hosts. Well, in fact, it's a prevalence where I divide it by the total population size. So it's the proportion of infected hosts through time. For an R node equal to 2, and S is the density of susceptible hosts, so those that are not yet recovered. And so this is in a linear plot, and then if you use log scale, then you see that indeed during the early phase of the epidemic there is an exponential growth, so you see a linear increase in the plot. And you can then play with the parameter of the disease. So for example, if you increase R node, you see that the epidemic is faster, so the slope increases like that, and you get a bigger epidemic. And you see also that the drop in the number of susceptible hosts is faster. And at the end, you get this number, which is at the end when the epidemic is done. So you see the total number, the total size of the epidemic here, which is the cumulative number of infected hosts that got infected throughout the epidemic. Okay. So one thing to realize is that because here the model assumes that when you recover the individual, you're immune for life, it means that the availability of susceptible hosts keeps decreasing through time, which means that after some time the epidemic will go down because there's no more susceptible hosts to infect. Okay, so this is a typical parameter values for the COVID-19, and now we can play with the effect of social distancing. So this is without social distancing, and then we can increase the parameter for social distancing, and we're going first to assume that this social distancing is permanent. When it's put forward, we're going to assume that it will be maintained forever. Okay, so the gray shading indicates the time at which we initiate social distancing. So those are the parameters for social distancing. And you see that when you do social distancing, it indeed affects a lot the dynamics. So it flattened the curve. So that's people have been saying a lot over the news. We want to decrease the epidemic size, and it will slow down the epidemic. So if you even further increase the social distancing, you see that there is actually parameter values where you can actually decrease the epidemics to a point where eventually you will eradicate the disease. Okay. So next, you can use this model to look at the total epidemic size for different intensity of social distancing. Okay, so if you have no social distancing, you see that almost 90% of the population will turn up infected by the end of the epidemic without social distancing. And if you implement permanent social distancing, you see that social distancing will decrease the total epidemic size of the disease. And at some point, in fact, social distancing will actually decrease totally and will lead to eradication. Okay, so that's good news. So it means that it works. So we can manage the disease with social distancing. The problem with that is that maintaining social distancing forever is unfeasible, of course, for all the costs that economic costs that are attached with that. So what happens if we release social distancing after some time? So that's what I'm going to show here with this model. So here, that's the scenario where it's permanent. And we're going to stop social distancing at some time. So like 180 days after the start of the epidemic. And what you see for these parameter values is that if you release social distancing at this point, there's a lot of susceptible hosts that are around, which means that there is going to be a second wave of the epidemic. So it will delay this second wave, but it will be there. And interestingly, if you use other parameters for social distancing, so for example, if you decrease the social distancing to 0.4, what you see is that there won't be a second wave of the epidemic. So why is that? Is that basically with this level of social distancing, there will be quite a lot of immunity that will build up during this phase. Okay, the density of susceptible hosts will decrease, will drop, which will basically decrease the opportunity for transmission at this point. Okay, so the total epidemic size actually turns out to be smaller in this configuration than when you have intense social distancing. Okay, so this is not completely intuitive. So to make this point even more clear, you can actually plot total epidemic size across the intensity of social distancing. So that was the previous plot I showed earlier, when you have permanent social distancing. And when you have transient social distancing, you get this curve. Okay, so you get a rebound here, which is due to re-emergence. So up to this point, there's no re-emergence, because if you reach this point at the end of social distancing, you have reached a level of herd immunity that actually prevents second epidemics. But if you go a bit beyond, then you incur the risk of having a second wave of epidemics. So so this graph shows that there is actually a level of social distancing, intermediate level of social distancing around 0.4 in this curve that minimizes total epidemic size. Maybe that's useful. The problem is that if you apply this rule, okay, you reach maximal prevalence, which is very high during this social distancing. And this remains unacceptable. So if you go back to this curve, what I mean is that if you apply this optimal strategy, you reach a point at the peak of the epidemic where 10% of the population is infected. So can we actually do better than that? So one way to do better is actually apply multiple bouts of social distancing, where in each bout you have a very strong social distancing. So that's one simulation where you can do that. And basically you manage to drop the size of the epidemic to a very low level. You release social distancing in the white window here. So maybe the epidemic will start, maybe you will manage to eradicate the disease by the end of social distancing. But if it increases again, then you can still go back to this strategy. Okay, and in fact, as time goes by, the herd immunity will build up, and maybe it will become actually easier to decrease the epidemic with time. So this strategy, multiple bout suppression, allows to decrease the maximal level of prevalence to a much lower level, which is good because maybe we can manage the flux of patients into hospitals by applying this strategy. It could lead to eradication because the strategy is very strong in terms of cutting the transmission. And also it will buy some time to develop drugs and vaccines. So that's it for the epidemiological model that we've been working on. And then I'm going to discuss what we've been doing on the evolution of the virus. Okay, and to study evolution, what we mean by evolution here is the evolution of phenotypic traits of the disease. So we need a slightly more complicated model. So here we still use the SIR model, but then we are going to consider different classes of individuals. So there is going to be an exposed class, so susceptible that become infected by the disease enter this class, which means that they are carrying the virus, but they cannot transmit the disease. And then at this stage, they can answer either a severe case, an infected case, or an asymptomatic case. Okay, so those parameter pi are going to determine the fate of the infection. So this is the first trait that we're going to look at, severity, which is going to control the probability to end up into the severe class. Then when you enter the infectious class, maybe the virus can modulate its transmission. So there are actually 3 different transmission rates associated to each type of hosts. And the virus may evolve different transmission rates in these different class of individuals. And then perhaps most importantly, we can also allow the virus to modulate its mortality rate in the severe case. So there's yet a third parameter that we can allow the virus to evolve. Okay, so and we also have a version where we allow for different types of hosts. So maybe some hosts are more sensitive than others, like older hosts have a higher probability to end up in the severe case. So we can, play with that. So, but I'm going next to focus only on this first version of the model. Okay, so to study evolution, and actually what I mean here is not evolution, but really adaptation. So whether the parasite is able to evolve a new phenotypic traits under natural selection. So we're going to assume first that there is a mutation that occurs in the virus population. This mutation is going to affect the phenotype. So it could be either the probability to end up in a severe class, it could be affecting the transmission rate, or it could affect the mortality rate. And the question of evolution that we're asking here is that we want to predict the dynamics of the frequency of the mutant. So can we say something about the change in frequency of the mutant? So it turns out that you can actually analyze this model and you end up with this kind of dynamics. So this is the change in the frequency of the mutant through time. And you recover some classical effects. So for example, this change in frequency depends on the availability of variation, so genetic variance. So of course you need a mutation for this adaptation to occur. And then there are 3 terms here. So the first refers to the evolution of direct selection on the severity. The second is about transmission, the evolution of transmission rate, and the third is about the evolution of virulence. So this equation shows that there is direct selection on severity, so the proportion of cases that are going to end up in a severe case, if there is variation in reproductive values among hosts. So those quantities here are reproductive values of the different types of hosts. And to give some intuition of what those quantities mean is that the reproductive value in this model is the measure of the quality of the host for the virus. So if, for example, severe cases transmit a lot of infections, it means that it's a good host for the virus because it will be a good vehicle to transmit to new susceptible hosts. Okay, so if it happens that the reproductive value of severe cases is very high, there will be selection for ending up in those severe cases. Okay, so there will be direct selection on this trait. This term here shows that there is direct selection on transmission. And in fact, it's a very strong direct selection because it's weighted by the availability of susceptible hosts. Okay, and during a pandemic, there's plenty of susceptible hosts available. It's a new disease. We humans are not immune to this disease, so it means that selection is going to be very strong to increase transmission rates. And the third term shows that actually direct selection is going to select against mortality, and that's a classical thing, result of models of virulence evolution. I mean, the parasite doesn't benefit in any way from killing its host, so that's a direct selection on mortality. Yet, there could also be indirect selection. So if you imagine that there are some trade-offs, so maybe if you are more virulent, then there is a higher transmission, then there is possibility for more deadly virus to spread due to indirect selection. So, but this requires trade-offs between all those phenotypic traits. And this equation is also useful to see what are the effects of social distancing on those quantities. So, first thing you can notice is that higher social distancing on beta is going to reduce selection for transmission. Okay, so if you apply social distancing, there's going to be a lower selection for transmission. And another thing you can do, so if you play with the social distancing via quarantining individuals, for example, if you quarantine severe cases, means that the reproductive values of severe cases are going to drop, and which is quite a good thing to do, because then it will drop those terms here associated with these severe cases. So selection, direct selection to end up into severe cases is going to decrease. So next, I'm going to show some simulation results. So this is a scenario without social distancing. And here we allowed a mutation. So this is to affect only transmission rate. Okay, so it allows to go from 2 to 2.5 in this case. And the dotted line shows the dynamic, the epidemiological dynamic through time without social distancing. So there is an exponential increase, and then the epidemic drops because of herd immunity. And if there is evolution, you see that's the full line. Because of selection for transmission, there is a higher transmission rate. So it's more than exponential increase in the epidemic. The peak of the epidemic is slightly higher, and then the epidemic drops sooner than without evolution. If you look at the mutant frequency, so here we started from 1% initial frequency. You see that there is very strong selection for transmission, and then as the number of susceptible hosts drop, the strength of selection for higher transmission drops also, but yet the frequency of the mutant keeps increasing and end up to almost fixation by the end of the simulation. So now I'm going to show a situation where we apply multiple bouts of social distancing. Okay, so what do we see here is that, okay, so if we look at the dashed line, so this is the case without evolution, you see that social distancing is working as indicated before. So drop the density of the infection, it increases, but then for the next drop, it keeps decreasing. But with evolution, because of the evolution of higher transmission rate, you see that actually the social distancing is failing in this scenario because of the evolution of the parasite. So it's a case of evolutionary rescue, if you want, because the parasite, by its adaptation, basically escape the extinction due to the social distancing. And if you look at the change in the mutant frequency in this scenario, so this is the black line. So you see that during the bout of social distancing, there is very strong decrease in selection for transmission, which is what we predicted. But if you release social distancing, the slope of this curve is actually very, very high. And why is that? It's basically, here you still have a lot of susceptible hosts available. And because of the increase in the mutant frequency, the genetic variance is very large at this point. So that's why there is a very steep slope. And in fact, with social distancing, this scenario shows that the mutant frequency is actually increasing faster than in this case, which is indicated as a dotted line. So the frequency of the mutation should increase faster than without social distancing. Okay, so just a reminder, those are completely based on, there's no data on those values. So here we're completely speculating about what could happen under some assumptions that we are just making up. So I'm not sure what's going to happen. It's just that in this scenario, we see these things. Whether or not evolution is going to happen, I mean, we know evolution is actually occurring right now. So we've heard just before that there is about 2 mutations per month occurring here, so which leads to this huge amount of variation among the viral population. The problem is that it's very difficult to know whether or not there is adaptive evolution, whether or not the phenotype of the mutant, of the virus is evolving. And it's very difficult because it's very difficult to decouple these types of pattern from the effect of founding events due to drift and regional differences between countries. Yet there are some results that are very strange. So for example, the mortality rate in Germany is supposed to be very small at the moment, whereas in Italy it's very high. So maybe there is variation in phenotypes that is hidden. And just to end, I'd like to make a comment about what happened in Ebola. For me, it reminds me a lot of what we've seen a few years ago during the emergence of Ebola in Africa, because the same kinds of questions about whether or not the virus was evolving were emerging. So, and these 2 papers were actually very nice because they arrived later on. During the ongoing epidemic, during the epidemic of Ebola, many people were actually sequencing the virus, seeing a lot of mutations, but nobody could actually determine or find adaptive mutations. And those papers actually looked at one set of mutations, so which is a mutation that appears in the glycoprotein of the virus, which is on the outer surface of the virus. And those are the maps of the frequency of the virus through time. So this mutation was first detected early on, and then it seemed to invade the population. Okay, so those are the orange regions in Africa. And but it was difficult to know whether this was the result of stochasticity or whether it was due to selection happening during the epidemic. So what those researchers did is that they basically did an experimental study. So they introduced this mutation in the virus, and they looked experimentally at the ability of these different variants to infect different immune cells of humans. And they were able to demonstrate indeed that those mutations that were detected in the epidemic were actually increasing the infectivity of the virus. Okay. So I'm just talking about this because it's an example where, in fact, phylodynamics was not able at that time to detect signs of adaptations, but those demonstrations came later due to experimental evidence that those mutations indeed increase infectivity. So maybe that's the same story could happen with COVID-19. So just to conclude, so the first part shows that maybe the multiple bouts of social distancing is a good strategy because it will reduce the peak of the epidemic, which is what is actually causing a lot of anxiety and disease and death. It will buy some time and slow down the epidemic and reduce also the total epidemic size and could even lead to eradication if all the countries are applying this at the same time. Regarding evolution, again, I'd like to be very careful that what we showed is without any data. So we need data to support any claim about what could be going on right now. But the model shows that there is strong direct selection for higher transmission rates. Okay, so if there is a variation in transmission rate, it's very likely that those mutations will increase in frequency because there's a lot of susceptible hosts around. There is direct selection against virulence. There is possibly direct selection on severity. There is a variation in reproductive values between hosts. And of course, if there are trade-offs between all those traits, there might be indirect selection on different phenotypes. So this evolution, in particular the evolution of transmission rate, could lead to an evolutionary rescue. So this is bad news. The good news maybe is that those kinds of models, qualitative models that we're developing, could maybe suggest some strategies to, for example, decrease the reproductive values of severe cases that could help to reduce the evolution and reduce the mortality of this disease. Okay, thank you.
Okay, so thanks, Sylvain. Let's see. Okay, so now we can move rapidly to the questions. So again, we're running really late. Apologies, everyone. This is like not only the first time we're having these webinars, but also we have 3 speakers, and we really don't even feel like interrupting them. I think you'll agree to that. So thank you to all the speakers. So what I'm going to do now is go through a link that Alex sent and try to, these questions have been rated. So for instance, there was for the first talk by Mircea, there was a question by Jesse Abati, about whether the model includes transmission from people who never develop symptoms, and also are you integrating data from exhaustive testing efforts?
So maybe all the speakers could switch back on their microphone and their cameras so that we can see them, and give us your thoughts.
And the camera. So Mircea, the first question, I started by a question to Mircea. He was the first speaker. Mircea?
Yeah. Are you hearing me?
Yeah. Do you see the question also?
I see the question. So thanks, Jesse. So first of all, there is no, this is not our model. What I understand from this is that is an issue that people can transmit while they are asymptomatic. Well, the fact that generation time is estimated through serial interval, but serial interval in fact is just a proxy for that. So this is not really important that this is linked to symptoms, because it is shown statistically and mathematically that they have the same expectation. Therefore, the estimate of serial interval doesn't depend on the clinical status. So what is important is not if there is a proportion of people that are asymptomatic and that they transmit the virus while they cannot be detected, is the uniformity of sampling and detection effort through time. That is that we have the same proportion of testing through time, so that the same we have the same proportion of expected cases among the whole confirmed cases among the real number of cases. So the problem is that one rather than the other. But while we apply this procedure to several countries, we see that the R0 is always ranging from 2 to 3.
Okay. There was another question that's both for Mircea and Sylvain, but I guess Mircea can still answer this one perhaps. Can we play on the network structure to reduce the spread, get more efficient or less costly social distancing by choosing whom interacts with whom?
Yeah, so this is a public health problem, and that requires logistics. But sure, the thing would be to reduce as much as possible the interactions between the susceptible and the recovered and infected individuals. That means that that would require to test all the people about their serology and find out which one are recovered. Then we assume that they are immunized for a while, and to isolate the and then they can do whatever they want because they are not susceptible and they are not infectious. And those that are not infected yet and are at risk of complications of critical disease, then those guys should avoid the contact. So that would require some kind of stratified lockdown, and that is, I think, the next step of lockdown in France.
Okay,
so maybe I can add up something on that. So because so that's really what actually allowed the WHO to eradicate smallpox. So what they really put up was a strategy of vaccination that was very aggressive during the ending days of smallpox. And basically the idea was as soon as there was a case reported, people would go, a team of the WHO would go and vaccinate all the population around this village, usually. And that's a very good strategy to actually target specific cases like that. I mean, the easy thing with smallpox is that there was a lot of symptoms. So each time someone is infected, there was a lot of symptoms. The problem with this disease is that there's a lot of infections that are hidden. So to basically be able to implement that in this case, you would need a lot of ways to test individuals. So that's one difficulty I see with this virus.
Okay, so well, Simon was adding some further comments, but I think we can discuss. You can discuss this perhaps afterwards. And I'd like to move to. There's a series of questions to Nicolas about whether the mutations have been characterized, whether the mutation rate differs in different regions of the genome, etc. Do we know something about their perhaps their functionality? So I don't know if you're aware about any of this, Nicolas.
Yeah, actually I skipped the last slide, and the last slide was all the mutations that were mapped. You had,
so perhaps you can show it now while we talk.
Well, you had non-synonymous mutations and synonymous mutations. They are quite rare to date, so it's difficult to say something. People focus on the S protein, which is the one expected to evolve quickly. But to date, there's nothing really clear. I should also say that I said you have 2 mutations per month. It's the molecular clock. The mutation rate is estimated to be 5×10⁻⁴. Okay, so it's quite a low mutation rate for a virus. So what people are discussing is that you have the strategy of the influenza or VIH virus that evolve very fast to escape the immune system. This is not the strategy of the coronavirus. Maybe the strategy of the coronavirus is to make immunity that lasts for a short time. You do not stay immune for long, and so it can reinfect again the same host. So it's a different strategy, and also people are arguing that as the population is not immune yet, you cannot have antigenetic drift, which is the kind of adaptive evolution people are tracking in virus. But what people don't discuss a lot, and I was very interested by Sylvain's talk, is evolution of life history trait of the virus. And it's really possible. You have the genetic diversity because the population size is very high. The mutation rate is lower than other viruses, but you have high genetic diversity. So you have everything needed on the side of molecular evolution to select for different life history traits, especially that this virus is new and it did not have time probably to improve the best strategy for a human population. And finally, so people ask if mutations are in some region of the genome, and yes, it is. So again, it's in the S protein, you have much more amino acid variation than in other parts of the genome.
Okay, thanks. Actually, the highest rated question is, do we have an estimate of the level of social distancing imposed by current strategy in France? Probably for Sylvain and Mircea, perhaps. Do we have any idea of what are the parameter values that are relevant?
No, there is no such an estimation. Those will be given in the next days, because we account between 2 and 3 weeks for the whole cycle of, in average, for the disease. But we have to take into account the death that can be delayed in their reporting. So that would lead us to mid-April to have an expectation of this.
So sorry. So in practice, you will use the change in the epidemiological parameter to infer this.
So what's happening is that I tried to stay into the 15 minutes, so I had to skip some slides, but it was another parameter that was interesting to show was the what is called the effective reproductive number. It's the basic reproduction number accounted for the decrease in the susceptible density. And this can be assessed through using several time-varying reproductive number by moving a time window and averaging on that. And so we have seen that this has already has an impact in South Korea, for example. They started with R0 about 2.5, and then now it's around the 1.5. So we know that this can happen, and this can be measured. For the lockdown is too recent for France, so that we cannot have an estimate for that. However, models such as the Ferguson team at Imperial College in London, they assume that the decrease can be up to 75% of the contact rate. But this is difficult then to relate to the situation in France and to the effect on cases, on incidence.
Okay, so we're going to finish pretty soon, I think. There was one question that I think is also interesting, perhaps for Nicolas. Someone is wondering, Sumanta is wondering if different virus strains are responsible for the target age group. In the US, it seems that the COVID hit younger people, while in the EU, it's mostly older people. So do we see something about genetic structure among age classes or these types of things? Do we know anything about this?
I'm not aware of any results, but in the Nextstrain tool, you can have the age of people. So it's possible to investigate if you have something. But I'm not aware of this.
Okay, so just one question for Sylvain. Sylvain,
yes.
Okay. So someone is asking if your model behaves differently if different countries have different policies or timing of social distancing. So I guess this would be something like including some spatial structure with delayed timing or different strategies. It's a different model for sure, but can you have an idea about this?
So we didn't allow for spatial structure. I mean, this would involve a lot of modification and complexity, but for the timing of the start of the social distancing, that's something it's easy to implement in our model. And it's hotly debated among countries. And so we wanted to explore that. And indeed, something you can see is that if you delay social distancing, you will allow a lot of herd immunity to build up. And of course, it's going to be very costly on the population because many people are going to be infected. Some will be severely infected, some will die.
So that was the original UK strategy, right? And
that was the original UK strategy, and that's why also
Denmark, perhaps, or
yeah. But indeed, if you do that, or if you fail to put up the social distancing very fast, then you will benefit from that. There will be a benefit for the rest of the population. The benefit being that there will be more herd immunity, so the effective reproductive number will be lower, and indeed you will be able to, you have more chance to decrease later on the spread of the epidemics, because you will use that. It will decrease the transmission of the disease.
Okay, so I guess we could stop now because we're it's already 1. Perhaps everyone feels hungry. So really, on behalf of the whole committee, which I think Emmanuel can open up his mic again, or I really want to thank all the 3 speakers. It's really a great effort. We asked from them to sort of put something up in a bit more than 1 week, and they really played it well, and I think everyone was really interested, judging by the questions. So we'll have the questions stored here, so you can think about them and perhaps try to answer them later. Then we recorded everything, and there's a chance we broadcast this later a bit more broadly. So we filled up the 500 attendance we were allowed. So that's pretty interesting. So thanks everyone, and
thank you.
Thank you.
Thank you, everyone. And well, you know, this is a weekly seminar series. So if you're interested, we're going to be back next week.
It won't be about COVID this time. It will be regular ecology and evolution. All right.
Thank you.